A blessed noon, everyone. I am Margie Rizal Giupay, and I'll be spearheading the presentation prepared by Group 7, which will be tackling about the protein synthesis, which later on will be deciphering the five main steps, which describes the protein synthesis in a basic or complex manner. So for this noon, I'll be having a quick introduction and we'll proceed directly to the protein synthesis, which is our main point for this uh, lecture. So we have three learning points that we'll be discussing in this presentation. The first learning point is of course, the overview of our protein synthesis, which I'll be discussing a little bit of a main concept, which is the central dogma of biology, which is quite really helpful insight or illustration for us to really understand what is the replication, the transcription, and the translation phased. So for the second learning point, we have the transcription, which is the RNA synthesis. It has a three subparts. We have the post-transcription, the genetic code, and the anticodons and the tRNA, which was already discussed in our previous discussion last lecture with Sir Jason. For the third learning point is the translation, which is the protein synthesis, which we will be discussing the five main steps, which is the activation of the tRNA, of RNA, tRNA, my mistake. And next is proceed. The second step is the initiation. The third step is the elongation. The fourth step is the termination. And the fifth and last step is the post translational processing. So guys, di ba, we all know that protein synthesis is the process by which our cells build proteins. The molecules that do most of the work in our body, it happens in two phases, in which the first phase is the transcription phase, in which you can see the diagram, which is DNA to RNA is transcription and RNA to protein is translation. That's the basic illustration that you can engrave in our minds for us to really understand what's the basic concept of the protein synthesis. So again, the first phase is the transcription, in which the DNA is copied into the RNA inside the nucleus. And the second phase is the translation, in which our DNA is somewhat decoded to make proteins in our cytoplasm. So as you can see in the diagram, it is a simple lang, a basic overview of how it flows from DNA to RNA to protein. So this process is summarized by the central dogma of biology, in which this is a very, very simple illustration in which from DNA to RNA to protein. Think of it like this. It's like DNA is our recipe book, and somewhat the RNA is like the photocopy of that recipe. And once the proteins are and once it is copied, like we copy from that recipe, so the proteins are somewhat our finished these dishes that our body needs. So as you can see here, from replication, we could see the DNA to DNA, and then by transcription from DNA to RNA using the RNA polymerase. And for the DNA replication, we use the DNA polymerase. And lastly, for the translation, it's the conversion already of RNA to protein, in which it is the protein, the how do I say this? The place where the protein synthesis take place is, may I remind you, is the ribosome. And in that specific place, there is a protein that is the final product. So, moving on, which is the transcription or the RNA synthesis. So, transcription happens again in the nucleus. Here, it's a section of our DNA in which it unwinds, and only one strand is used as our template. So, the RNA polymerase, it somewhat lang builds a complementary RNA strand called the hnRNA, or the heterogeneous nuclear RNA. So this RNA isn't ready, guys, may I remind you, because it still has extra pieces. You just have to remind yourself that hnRNA has to be converted pa into mRNA. That's where we'll be going to discuss later on, which is the post-transcription. So for the post-transcription processing, as you can see here, the hnRNA, which is the heterogeneous nuclear RNA, is converted na already to mRNA in the post-transcription processing. So the RNA produced from a gene to a transcription is hnRNA. So the precursor for the mRNA is of course the hnRNA. So the conversion of the hnRNA to the mRNA, it involves a post-transcription processing of the hnRNA. As you can see here in the diagram, it's somewhat complicated. Just focus on the terms, which is exon and intron. Exon is a gene segment that conveys or codes for a genetic information. Intron naman is a gene segment that does not convey or code for genetic information. So that's our introns. As you can see here, the entrance is somewhat removed. It's been cut. And then once it has been removed or cut, the exon has been glued or like sumpay, nagsumpay-sumpay. So that's already the final product, which is the mRNA. So as easy as that, the exon is the one that carry our genetic information, and tron is the one that does not carry our genetic information, that it has been spliced or removed. And then once the exon has been connected to each other, then we can form the mRNA. So hnRNA must be processed into mRNA, diba? Because mRNA is very needed sa atong translation process before leaving the nucleus. So genes are of course made of exons, which is the coding part again, and then for the introns, that's our non-coding part. So both exons and introns are copied sa atong hnRNA, but atong introns are removed through a process called, I've mentioned it earlier, that's splicing. So the small nuclear RNA and proteins together, called the RNPs or the SNRPs, form the spliceosomes. They are the one that cut out, as you can see in the illustration, that cut out the introns and allowed the exons to join with each other. So the final edited strand, that is our mRNA. So which carries the actual genetic message to the ribosome. So you just have to remember sRNA, the small nuclear RNA, that is the one. That is the RNA that facilitates the conversion of the heterogeneous nuclear RNA to the mRNA. So, di ba, we already have discussed the different types of RNA. We have the heterogeneous nuclear RNA. We also have discussed the messenger RNA, the small nuclear RNA, the ribosomal RNA, and transfer RNA. So, you just have to remember that five RNA types because it is a very important or crucial elements in the protein synthesis. So as I've discussed already, the snRNA facilitates the conversion of our heterogeneous nuclear RNA to the mRNA. So moving on sa post-transcription, of course, as we go along the translation, we always have to make sure that we need to discuss the genetic code. So in the post-transcription processing or formation of our mRNA, we have the genetic code. So the mRNA is written somewhat in a language called the genetic code. It's more on like mo siya atong alphabet ba in terms of genetic coding. So each codon is set of a three nucleotide. It's like a three-letter word. If you observe, the left most portion part is the first letter. The upper part or the center one is the second letter, and the rightmost portion part is our third letter. So each codon is set to a three nucleotide. It's a three-letter word. With, for example, with four nucleotides, you can form uh, AUCG. So there are 64 possible codons. Again, there are 64 possible codons, but 61 codons are code for amino acids because, again, they are the other three, the remaining three, is act as stop signals. So just focus on the highlighted blue part ish here. You could see the first part here in the leftmost portion is we have the initiation codon, which is the methionine. It's AUG. And to the rightmost part above, you can see the uh, highlighted blue part. We have the UAA, UWAG, and UGA, which is our three stop codons. Again, the 61 codons code for amino acid, and the other three act as stop signals. So for the AUG, that's the initiation. It's also called the methionine. For our three stop signals, again, may I remind you, it's UAA, UWAG, and UGA. So this fun fact, this was one of the greatest discovery in biology because they were able to decipher the genetic codes, which somewhat earned a Nobel Prize before in 1968. It's a very famous and monumental discovery before. Next is the anticodons and tRNA. So before proceeding to translation or protein synthesis, I have to discuss anticodons and the tRNA molecules. Again, may I remind you that tRNA is, di ba, the transfer RNA, in which it is the RNA that delivers our amino acids to the site for protein synthesis. So transfer RNAs are quite the smallest RNA process. It only possess around 75 to 90 nucleotide units. So again, proteins are not built directly by mRNA codons. Instead, the tRNA act as the translator, or somewhat it transfer. So each tRNA carries a specific amino acid, guys. So at one end, kaning tRNA, it has attachment site for its amino acid. You can see here, diba? It's like a cross, or just imagine it as a cross ba, or mura ba siya hairpin for girls, if you try to illustrate it in a different analogy. So at the other end, it has an anticodon. Kanang sa cross part, the bottommost part, mura siyang tail niya, it has a sequence of three bases complementary to an mRNA codon. Again, that is our anticodon. So when the anticodon matches with the codon on the mRNA, so the correct amino acid is placed into the growing protein chain now. So think of it like si tRNA is like our delivery track. So the anticodon is our delivery address, and the amino acid is our package. For instance, the delivery address is Southwestern FEMA, and our package is the amino acid. So once it will be arrived, then of course we can assume that the amino acid is already have been received or placed into a growing protein peptide chain. So moving on to that, the interaction between the anticodon of the tRNA and the codon of the mRNA leads to what? The proper placement of our amino acid into a growing peptide chain during the protein synthesis. So the interaction, of course, involves a complementary base pairing. So just have to remember the terms amino acid. I know you're not quite familiar with amino acid. Amino acid is mo na siya ang ma-form from the 3' end of the amino acid, and then the carboxyl of our amino acid. So the amino acid, diba, is carboxyl. Our 3N is OH. So when they combine, you form an amino acid. So moving on, of course, we are now here in the translation and the protein synthesis. So, which happens again in the ribosomes in our cytoplasm. So the ribosomes acts somewhat like a factory, which it reads our mRNA instructions in assembling amino acids into a polypeptide chain. So the translation has a five major steps. We have the activation of the tRNA. The initiation is the second step. The third step is the elongation. The fourth step is our termination, and the fifth step is a post-translational processing. So I'll give the floor to my members who will be explaining the different side of the different steps of the translation or the protein synthesis.
Let's talk about the activation of tRNA. This process is the very first step in protein synthesis. It ensures that each amino acid is attached to the correct tRNA before being delivered to the ribosome. Here's how it happens step by step. First, the enzyme called aminoacyl tRNA synthetase recognizes a specific amino acid and its matching tRNA. Next, the amino acid reacts with ATP to form aminoacyl AMP. Then, the activated amino acid is transferred to the 3' end of the tRNA, and AMP is released. Finally, the result is a charged tRNA, also known as aminoacyl tRNA, which is now ready to deliver its amino acid to the ribosome for protein synthesis. In short, activation of tRNA means linking the right amino acid to the right tRNA, with the help of ATP and aminoacyl tRNA synthetase, to ensure proteins are built correctly.
So here we have step 2, the initiation phase. In initiation phase, this is the starting phase of translation, where the cellular machinery comes together to begin making a protein. So it involves the small ribosomal subunit binding to the mRNA start codon, the AUG, followed by a special tRNA carrying the first amino acid, methionine, which binds to that codon. Once the large ribosomal subunit attaches, the complete ribosome is formed and ready to begin making the protein.
Elongation is the second step of transcription. Here, RNA polymerase reads the DNA template and builds an RNA strand. It moves along the DNA in one direction and adds RNA bases that match the DNA bases. A pairs with U, and C pairs with G as it moves. It opens up the DNA ahead and closes it behind. The new RNA strand grows longer as more bases are added. This continues until RNA polymerase reaches a stop signal. Last step of translation, the termination step. It begins when a stop codon is read. There are 3 stop codons you need to be familiar with. First is UAA. Next is UAG and UGA. These stop codons, also known as nonsense codons. They don't code for any specific tRNA molecule. Instead, when these are read at the A site, they cause a release factor to enter the A site, which causes the small and the large ribosomal subunits to basically disassemble. So this is where translation ends and the protein leaves the ribosomes, where it eventually goes in the Golgi body for further processing and modification. At the Golgi body, the proteins undergo folding, where they form a specific shape to perform a specific function.
So the last step is the post-translational processing. So post-translational process, it refers to the modification that occur to a protein after it has been translated from messenger RNA. Although nucleic acids such as DNA and RNA direct protein synthesis, the post-translational process ensures that the resulting protein is folded, cut, chemically modified, and activated so it can perform its correct function in the cell. In short, it is the processing of proteins after translation of nucleic acids, turning raw polypeptides chains into functional proteins.